Hello everyone, I'm Jimena Ibagon, a first-year PhD student, and you are tuning in to the Enhanced Age-Sustained Monogastric Nutrition Laboratory podcast. Today's episode is a deep dive into my research on the effect of microbial phytase on the digestibility of phosphorus in 7 sources of sunflower co-products fed to growing pigs. This research was part of a poster presentation I shared at the Midwest meeting of the American Society of Animal Science in 2023. To give you a bit of introduction, the nutritional composition of sunflower co-products can indeed be influenced by various factors, such as the location where they are grown, the degree of the dehulling they undergo, and the method used for the oil extraction. These factors can impact the levels of phosphorus, calcium, and other nutrients present in those co-products. Furthermore, the inclusion of microbial phytase in pig diets has been shown to enhance the digestibility of phosphorus in other oilseeds. However, it remains unclear whether the geographic region where the sunflowers are grown, the technique used for oil extraction, have an effect on the digestibility of phosphorus and calcium in sunflower milk co-products when these are fed to growing pigs. Understanding these relationships could lead to optimize the feeding strategies for swine nutrition and improve utilization of sunflower co-products in pig diets. Therefore, the objective of this experiment was to test the hypothesis that there are no differences among sources of sunflower co-products in the apparent total tract digestibility of phosphorus and calcium and on the standardized total tract digestibility of phosphorus regardless of where the ingredients were grown. In these experiments, we used 7 sources of sunflower co-products. This includes 6 sources of sunflower meal, 2 from Ukraine, 2 from United States, 1 from Hungary, 1 from Italy, and additionally, we have 1 source of sunflower expeller from the United States. 7 diets were mixed, where sunflower co-products were used as the sole source of phosphorus. Furthermore, another set of 7 diets were mixed, identical to the first 7, with the exception that those diets contain 300 units per kilogram of microbial phytase. With that, we have a total of 14 diets. A total of 112 burros were randomly allotted to these 14 diets, and those pigs were placed in individual metabolism crates with slatted floors that allow us for total but separate collection of urine and fecal materials. Now let's move on to the results, and I would like to set up my graphs. We have 7 bars, and each bar represents one of the sunflower co-products. And in graph 1, we observe that the apparent total tract digestibility of phosphorus stands out, that one of the sources from Ukraine and the source from Hungary show us a greater apparent digestibility compared to the sunflower expeller, and no significant difference were noted among the other sunflower meal sources. An interesting observation was noted and was that adding the 300 units of phytase in the diet significantly increased the apparent total tract digestibility of phosphorus across the sunflower sources. This addition seems to enhance the overall digestibility of phosphorus in all the sunflower co-products. Moving to figure 2, a similar narrative we observed, this time around the standardized total tract digestibility of phosphorus. What catch our attention is that within sunflower meal sources, no significant difference in the STTD of phosphorus were noted among its sources. However, the standardized total tract digestibility of phosphorus in the sunflower expeller was notably lower compared to the sunflower milk co-products. Additionally, the inclusion of phytase show us a consistent trend emerged across all sunflower co-products. This means that an increase in the standardized total tract digestibility of phosphorus in all sunflower milk co-products was observed. Now let's focus on figure 3, and this graph is about the apparent total tract digestibility of calcium, and we observe that one of the sources from the United States have a higher digestibility of calcium compared to other sources like Ukraine or Hungary. However, no significant difference were evident among sunflower meal sources from the United States, Italy, or the sunflower expeller co-product. And the addition of the 300 units of phytase to these diets contribute to an increase in the digestibility of calcium across the sunflower co-products. In conclusion, the addition of microbial phytase to pig diets containing sunflower meal or sunflower expeller show us a consistent outcome, an increase in the digestibility of calcium and phosphorus regardless of the type of sunflower co-product used. The difference observed in the phosphorus digestibility among sunflower co-products within this experiment could be attributed to variation in growing conditions or difference in the processing of sunflower seeds for oil extraction. With that, I would like to thank you for tuning in to this episode. And if you have any question, feel free to reach out via email to jaibagon@illinois.edu or visit our website at nutrition.nc.illinois.edu. Until next time, and please stay curious exploring the fascinating world of animal nutrition.